hairpin peptidomimetic drug.
Right, so representing the design of a beta-hairpin peptidomimetic drug. We'll start by giving a brief recap of protein epitope mimetics, followed by a look at our drug target. Then we'll go over some of the options for synthesis design options for our molecule, and then we'll look at the synthesis methods as well as evaluation techniques. Right. So, what does protein epitope mimetics consist of? Most drugs and drug-like molecules tend to be limited to very small regions of molecular space, and they follow very strict guidelines, such as Lipinski's rule of 5. And the problem with this is that it leaves many diseases and illnesses without many treatment options available, because the proteins responsible for them are not amenable to small drug-like molecules, but rather they are regulated by protein-protein interactions, which usually translates into larger and flatter surfaces. However, it's a good starting point for structure-based inhibitor design. And this is where protein epitope mimetics comes in, and it consists basically in imitating the epitopes of biologically relevant proteins using small peptide sequences. And a very important structure for PEM is the beta-hairpin. It's a naturally occurring motif in proteins and peptides, and it's present in the epitope, in the antigen binding sites of antibodies, as well as in the ligand binding sites of several proteins. And it possesses a high degree of structural diversity due to variations in loop size as well as amino acid sequence. The challenge with this technique, however, is in stabilizing discrete peptide conformations, as well as ensuring that they survive long enough to have a therapeutical effect. For instance, because of proteolytic degradation. And on top of that, beta-hairpins require specific sequences or templates, such as the dipeptide D-proline, L-proline, in order to induce a turn and fold properly. However, fortunately, there are some options to address these issues, and there are two types of structural changes that might be incorporated for this. And the first one are cyclizations, which include consists of closing a loop in the peptide amino acid sequence in order to create a macrocyclic structure, which will add to structural rigidity as well as stabilize the conformation. And it can be linking the C termini and N termini together, or side chain linking, such as disulfide bonds between cysteine residues. And then we also have some backbone modifications using, for instance, beta peptides, which have higher conformational stability, or using the peptides, which have higher resistance to proteolytic degradation, despite having lower conformational stability.
Yes, and so we also selected a target to look at especially, and this is the CXCR4 receptor, which probably doesn't tell you much, but it's a G-protein coupled receptor, which means that, as you can see, this is supposed to be showing like the membrane of a cell. So it's a transmembrane receptor, and it has 7 transmembrane domains and 3 extracellular loops and 3 intercellular. So what happens with those receptors is that you have a ligand binding outside the cell, which trigger a signal inside the cells. I don't know if you this called like G signal parts that are released in a sequence. Now, this receptor is a part of the immune system and can also be found in the central nervous system. And also they are part of the hematopoietic system. And this is the system where stem cells are converted into blood cells. So it does do a lot of good things, but the problem is that it's also connected to the entry of HIV cells, or cold receptor therefore. It has also been shown to be overexpressed in tumor cells, where they are assumed to be contribute to tumor growth and angiogenesis, which is the growth of blood cells to support the tumor, and also drug resistance. And I will try to talk a bit about the receptor so that we understand what the pocket looks like and what we need to design our molecule. And so you can actually see a receptor up there, which is this cyclic peptide inhibitor. And in the PDB we have two crystal structures, and one is with a small molecule and one with this cyclic peptide. So there could be alternatives, although our mission is to create a beta peptide, which is much closer to this cyclic peptide. This pocket is polar, which is also important to remember when we're designing. And the natural ligand is SDF1 factor, and I will actually show you that in this slide. And I don't know if you recall what you saw just a few seconds ago, but this part actually looks a bit like a β-hairpin. So this might be a sign that this is actually something that could be used, but it's also already in the natural ligand. Just to get an idea of what the binding pocket looks like, this is our receptor seen from above. This is the small molecule, so and you can see the small molecule, and you can see that's very far down. And instead, this cyclic peptide is more covering a larger part of this binding pocket, and I think that's where we want to go, because with this, I think there's a less chance that we actually inhibit the receptor. So by covering a larger part, maybe that would be a winning concept.
And now let me introduce Atlantic horseshoe crab. Despite its name, this creature is more closely related to the scorpions, ticks, and spiders than to crabs. It has 10 eyes, 10 claws, and 14 legs, and blue blood that is highly valued for drug discovery. And scientists isolated polyphenylsin 2 with collecting its blood. This is the structure of the polyphenylsin 2. You can see that it is 18 amino acid long and is stabilized into amphiphilic and antiparallel beta-hairpin by 2 disulfide bridges. And this polypeptide shows an excellent activity against different gram-negative and gram-positive bacteria. The first synthesized close analog to the polyphenylsin 2 was T22. You can see that scientists changed non-polar residues as phenylalanines and valine to the tyrosine and lysine. The previous compound showed a potent inhibition of CXCR4. Further analogues, such as this one, are shorter, we can see that it is only 14 amino acid residues long instead of 18. You can see that there is 2 new residues like citrulline and 2 naphthylalanine. Citrulline is arginine-like isosteric analog, but it isn't charged. And you can see that now we have only 1 disulfide bridge. Also, scientists made a conclusion that the number of total +6 or +7 charges seems to be a suitable balance between toxicity and activity. And I think that these pictures are a bit terrible for understanding at first time. So I just showed them into this scheme. Polyfor is a Swiss pharma company, and they developed 5 different PEM molecules. But nowadays only 3 of them are
Used?
Now only the 3 are used now in different trials. And let's see how it was. So their starting point was T140, and you can see with red marked squares what had changed. So you can see that they started using our template D-proline, L-proline to obtain chemically and conformationally stable type 2' beta turn. And they use citrulline instead of lysine. Then they decided to cyclize the molecule to improve its stability. The next step was extending of the amount of amino acid residues. So you can see 2 additional amino acids and 1 replacement here. Unfortunately, the structure of pole 55-51 is unknown. There are a lot of articles about that compound, but the structure is unknown. I have contacted the Polyfor scientists for the structure of the molecule, but nowadays they are discussing whether to send or not the structure to our group. Nevertheless, you can see the difference between these 2 potent molecules. A lot of replacements, and now we use 1 uncommon amino acid, that is this one. And this is their structures. Thanks to protein database, we can download the structure of CXCR4 and CVX15 complex that you can see here. But don't forget that it's only a symmetric unit, and its unit cell consists of 2 asymmetric units. As we know, it's a 2-fold diet, and it has in situ symmetry. And now let's look at the bounding state of our compound to the protein. This is CVX15. It's an open analog to pole 3026. Usually we have some situation when we design peptide drugs. We have such situation like this. And as we have antiparallel β-hairpin, there are a lot of hydrogen bonds, and usually we have one residues that are turned to one side and others, and usually it's polar and non-polar. This one is turned to the protein and this one is turned to the solvent. But in our case, this concept doesn't work because our molecule dipped into the pocket of the protein. So we also have to think about different molecular interactions. It's very important. You can see hydrogen bonds with yellow dashes. And our conclusion about what we can do. We don't have to change to systems and our template. It's very necessary component of our compound, and these residues are relevant and may be very necessary because they form sufficient amount of molecular interactions, such as hydrogen bonds, and they feel excellent the free area of the pocket. So what can we change? Let's start from G-proline. You can see as we have cyclized form of G-proline, it doesn't form this interaction. So we can change it, and there are a lot of free space here. So we can change it to aliphatic, aromatic, or hydrogen bond donor residue. Hydrogen bond donor because if you look to the structure of the protein, it is close to hydrogen bond acceptor oxygen of the CXCR form. And you can see that adenine forms only one hydrogen bond to the protein, so we can improve it. Let's look at citrulline. It forms only one hydrogen bond, and according to its size and properties, we can replace it with these residues. And as to naphthylalanine confirms pp stacking, pi stacking, we can replace it using different aromatic residues. The same situation with this arginine, and if we talk about tyrosine, you can see that its aromatic ring is close to the aliphatic part of proline. So we can use different ways, change it to aliphatic residues or to aromatic one, and look if it will help us. Moreover, you can see that our suggestions are similar to the changes in this molecule, so we hope that we are on the right way, and our suggestions will lead us for discovering a new potent drug for inhibit CXCR form.
Right, so now we take a look at some of the synthesis options, and the best one would be solid phase peptide synthesis, and it is based on the concept of using FMOC protection groups for alpha amines in amino acids, and FMOC stands for N-fluoranyl methoxycarbonyl. And it's essential in this process, since it allows the chemist to control the reaction and add the amino acids in the right sequence. And it uses a resin to which the initial amino acid is added, and it uses a coupling reagent for this, such as HCTU, and then we use piperidine to remove the protection group, and this is done successfully until the final amino acid, until the final peptide is obtained, which is then cleaved from the amide from the resin with trifluoroacetic acid. And it's ideal for small-scale production and even afterwards to test the compound. And it's relatively straightforward, although there are some things to keep in mind. For instance, the use of resins with lower capacity in order to avoid reactions between different chains. And now this brings us to techniques to evaluate the outcome of our compound, and we have analytical techniques which are more relevant for structural analysis, and then biological assays to test the pharmacokinetic properties of our compound. And the first technique is MALDI, which stands for Matrix Assisted Laser Desorption Ionization, and here the sample is mixed with an organic matrix such as synapinic acid, and the matrix is then irradiated with a beam. This in turn leads to, this will ionize the matrix, which in turn will lead to sample ionization, and this technique can be coupled with mass spectrometry for analysis, and it has many advantages. It's a soft ionization method, so it usually is not destructive, and it also tends to produce ions with unitary charge, and this makes the spectra easier to interpret. Then we have other techniques such as NMR, and since short peptides are not prone to preserve secondary structure, and pore stability can decrease to binding infinity, it's important to ensure that the structure is adequate. And a stable beta-hairpin structure, which was adopted by the T22, was approved by NMR. And the arrows in this figure represent the long-range NOEs of the protons located in the interior of the beta sheets on the opposite beta strands, and they help confirm this structure. Then we have surface plasmon resonance, which is an advanced biosensor technique that enables measurement affinities and kinetic rate constants of ligand-protein interactions. And when integrated with other structural analytical work, structural kinetic relationships can be deduced for optimization of the lead molecule. Then X-ray crystallography provides a powerful tool for analyzing the binding mode of inhibitor, and the crystal structure of the ligand-protein complex can also reveal how the surface of the protein adapts to the hairpin, and thereby providing clues for enhancing inhibitor affinity. And this brings us to in vitro studies to analyze our molecule's ability to compete with CXCL12 at the level of receptor binding and correlation between binding and the relative potency of the compound. in order to inhibit the receptor signaling and function. The data obtained from these assays provide receptor inhibiting activity, but they also serve as future screening strategies for the identification of novel anti-CXCR4 drugs. And CA2 plus flux assays commonly reported in literature to analyze the binding potency of designed compounds on GPCR-related signaling. And the principle of the measurement is based on changes in fluorescence intensity or emission wavelength of a fluorophore binding to calcium atoms. And then IC50 was determined for the optimized analogues of P22 in calcium ion flux assay, and POL2438 as well as POL3026. Their stabilizing hairpin structures exhibit significantly improved affinities for the target. And finally, in vivo efficacy of POL306 has been assessed by murine colony forming cell assay, and improved circulating of hematopoietic stem cells was shown compared to standard GCSF treatment. And HSCs usually reside in bone marrow, but CXCL12 as well as and CXCR4 axis results in mobilization, which is very important for transplantation for treatment of many hematological diseases such as leukemia.
Yeah, I think we're almost exceeding our time, so I would just show you the summary of what we would like to do for this molecule. Thank you.
Thank you.
Any questions?
Questions? Yeah, Thomas was first.
This is just a clarification you can make. The funny animal that had a blue blood, can you explain more how this comes in? I didn't understand. Was it the producer of the first product of this class, or why was this animal so interesting?
This animal has this blue blood, and this blood consists this polyphenyleucin too. And this compound, when it was isolated, showed an excellent activity against gram-positive and gram-negative bacteria. So they wanted to design some antimicrobial drug, and then they saw that we can inhibit CXCR4.
This was a chance finding, I guess. That it was just coincidence or screening that they found that CXCR4 could be inhibited.
Yeah, it seems to be quite, what do you say, slumpen, or in English.
Yeah.
Just by default.
So it was the inspiration for the rest?
Yes,
It is.
it was the first starting point. Then.
It's a very common situation. Good example.
Yeah, Stefan.
The β-hairpin you draw looks good, but the non-polar and polar side of the residues, it's an up-down pattern, but not on the strands, but in each. And
So,
I understand.
so it's just you explained it right, but you drew it wrong.
Yeah, sorry.
So in an anti-parallel sheet, yeah,
Thank
so
you.
there are always pairs showing on the same side.
This one?
No,
Oh, no.
and
they're both up. They're both up,
Mhm.
and the next one are both down.
Ah.
Ah.
Yes.
Yeah, exactly. Up, down, up, down, up, down. Exactly, perfect.
Answer.
So there's results in if you have each second one a polar and each second one an apolar one, you have a polar and apolar side, which is more facing the solvent side and one more facing the pocket or surface side, as you said. Bigger molecules more bind to surfaces than to pockets.
Yes.
Also on the topic of the blue blood, actually the blue blood is due to hemocyanin, which is a type of our blood is red because of hemoglobin. This animal's blood, also some spiders' blood, is because of hemocyanin, not because of this compound. But this compound is found in that animal. So they, after they did the tests, they split the blood like you do normally in the lab. That's when they found the compound that they used to to inhibit or prohibit the target.
You mentioned here at the end risks of bad admit.
Yes.
What do you mean, or if I'm well, if I ask a general question, what do you mean there?
Right. Now it's gone, so we can't talk about it.
Yeah.
No, but what I mean is that as we discussed a lot with peptides or protein structure-based drugs, is that they usually have poor bioavailability and are degraded fast in the body. So I think that's a problem, that if you don't get it stable enough, or that it, what can I say, if it's too big, if you want to take it orally, say, then that would be like a risk with this project, trying to develop a drug that might in the end up not being compatible.
Would you even try developing this as an oral drug? If it was your money, your $1 billion, would you invest it in trying to give this as an oral?
Like no, because I am not a risk taker.
Yeah, no, but that's the right answer.
But I think, as I said, the polyphore is actually, I think it's clinical trial 1 or 2, one of these poll. So, and that's the structure that we couldn't
One
find.
is on the clinical trial 2 and 2 on the clinical trial 1.
Yes.
So someone is
But
maybe
I
on
think
their
they're
way.
injecting it, I would
Yes,
guess so.
probably,
And
yes.
that is fair. I mean, if you've got cancer, you're quite well prepared to come into the hospital and have an injection of a drug, or you may even be able to inject it yourself by a syringe pump, which you have sort of inserted, and you can dose yourself at home. So, I mean, oral absorption is good, but in some cases it's impossible, and then one just has to live with it and go for some other way of administrating.
But I think the company that really finds the compound in the end, I think they will get great economics out of that, probably. And also that if you know a structure that works, it probably won't be that difficult to change it, so that maybe you could get more patents out of it.
Yeah, yeah.
So it might be something. Yeah. So there's a lot at stake, but also a lot to.
Yeah. Yeah, Thomas.
You mentioned in the slides here large-scale production, but you didn't say so much about it. It's a classical problem when it comes to peptide and protein. Should you produce them with an organism, or should you produce them with chemistry? What is your preference here?
I think,
For large-scale production, using a recombinant organism usually would be the better option. In this case, there are many more things to take into consideration, because also sometimes there are backbone modifications, such as the use of dipeptides or beta amino acids, and it's not just any organism that can just do all the types of necessary modifications. So it's better if possible, but it's not always. It's not as straightforward as the solid phase peptide synthesis.
Do you know any solid phase peptide synthesis of medium-sized peptides that are as used as pharmaceuticals at large scale? What is the limit? Is it 10 amino acids, 20 amino acids, 40 amino acids?
I'm not sure if there's really a limit, but the more more amino acids are incorporated, the worse the yield of the reaction is. And I think maybe after maybe 20 or 30 it starts to go really down, or maybe way before.
There are some recent examples. There is one pharmaceutical which is I think 30 amino acids, 20 amino acids produced by some in polymers. So it's not impossible, but somewhere there is a limit. I mean, if you say 100 amino acids, you would better go for an organism. But again, if you have unnatural amino acids, it might not be so. So it's a classical problem that people always discuss.
Yeah, I think for us, these structures would probably be better made synthetically, because you want them cycling and you want to have different unnatural amino acids in there.
May I have a question? It's
Yes.
more of general sort. So now that we're talking about the organism that would introduce also the unnatural amino acids, like there is no organism that would like solely produce that in such a complexity, like using big amino acids, but there are microbes that use the peptides to build, proteins to build other peptides. Is this used like industry-wise to produce such peptides already, or is it just the thing we know?
Industry has an interest in that, I know, because when I was at AstraZeneca we set up a collaboration with a Scottish scientist who was working with that to make these kind of modified peptides enzymatically. If it's being used outside of research, and I don't know, for production, I don't know honestly. Maybe Thomas or no. Okay, thank you very much. So then we see each other at 9:15 tomorrow for 4 additional presentations.